Hello again, 112. This is Ms. Hook. Today we'll be looking at Chapter 9, the patterns of inheritance. So we're going to look at genetics and how they pass from parents to offspring. The first person you should know about is Gregor Mendel. Mendel was a monk, and I guess it would be Czech Republic now where he was from. Spoke German in the mid-1800s. So really we haven't known about genetics and the patterns of inheritance for very long. He became a monk because back then, if you wanted to do science research, that was the best way to do it. So he looked at plants. He looked at pea plants mostly. And he looked at how traits were passed on. He would take different breeds of peas, different colored leaves, and he would see what rates their offspring would have the different colors. Obviously, people before then had noticed that offspring were like their parents, but they didn't know how to predict traits being passed on, and Mendel did. So here's a pea plant that he looked at. They have lots of different traits and they're easily manipulated. And they can self-fertilize or you can fertilize them yourselves. So what he did was he did cross-fertilizations, which means he took the pollen from one plant and implanted it in another, so that those offspring would be half the white flower, half the purple. Yep. So he's taking the pollen, sending it over, and then you've got what's called the F1 generation. So first you've got the parents, and then their first group of kids or offspring is the F1. He also created true breeding varieties. So what does that mean? That means that they only had one trait. So if they were purple flowers, both versions of their chromosomes were purple flowers. He'd bred them together. He could see that their offspring were all purple. He knew that they had to be true purple. And then he would make true white. He'd breed white ones together, see that all the offspring were white. And you know that's true white. And then what he did was he did a cross, where he crossed the true purple with the true white, and look at the offspring. How many were purple? How many were white? And then he'd breed those offspring together. This is a reason we can't really do this work in humans. Don't really want to be breeding offspring together. He'd breed those together for an F2 generation, and he'd see how many were purple and white then. He also looked at different characteristics of the pea plants like seed shape, seed color, the flower color, the shape of the pod, and the pod color. So these are a lot of different traits that he could test to see what happened. He also looked at if they were terminal, like they did the flowers at the end, or axial, they broke off the main axis, and if they were tall or dwarf. So there are a lot of different traits he could look at. So first he would do the monohybrid cross, and that's what we were talking about before, where you'd breed like the true purple with the true white. Mono means one. Hybrid means the offspring of crossing two different characteristics. So like a liger is a hybrid between a tiger and a lion. A hybrid car uses electricity and gas, so it's a cross between two different things. So when he did that, the first generation were all purple flowers. So that told him that the purple gene masked the white gene. But he wanted to see what would happen if he crossed them again. So all these flowers have one gene for purple and one for white. And what he found there with the F2 generation was that 3/4 had purple, but 1/4 had white. And by finding these ratios, he was able to then predict what was happening. Okay, so the 4 hypotheses that he formed all work together, so we don't have to just say one is true. So his hypotheses that there are alternate forms of genes. So that's called alleles. So like the different forms, the different alleles, an allele for purple flowers, an allele for white flowers. And for each characteristic, each organism has 2 alleles. So we've already talked about in the last chapter how we have two copies of each chromosome, one from mom and one from dad, and those have the same genes, but they can have different versions. That's what he was thinking. So that on each of your chromosomes, you've got two alleles. They could both be for purple, one could be for purple, one could be for white. They could both be white. And gametes, those sex cells, only have one of the alleles. So that when they combine together, you've got two. And these alleles, these gene versions, can be dominant or recessive. So they can mask the other, or they can be masked. We're going to look at that. What Mendel did to predict offspring was he made what's called a Punnett square. So on the top, you have one parent's genetics. On the bottom, you have the other parent's. So each of these Y's is a gene, an allele, a version of that gene. Here and here as well. And what we do for each trait is we give it usually a letter. So this looks to be the pod color. And a capital letter means it's dominant. It masks the other. And the lowercase means it's recessive. So if you have a big Y and a little y, you'll see the yellow. You'll see the dominant trait. You'll only see green, the little y, if you have two copies of it. That's what makes it recessive. So what you do to make a Punnett square is for each allele, they've got a 50-50 chance of sending them. So on the top, you send over the big Y, and the bottom, the little y. And then here you go down, little y, little y, little y, little y. And what you have are the four possible results of offspring. And if you look at the top two, those have a big Y and a little y. So they would look yellow. The yellow masks the green, they look yellow. The two on the bottom, they have two little y's, they look green. So here, 2 out of 4 are yellow and 2 out of 4 are green. So we would say there's a 50-50 chance they would be yellow or green. And that's going to depend on what this parent sends. Okay, so here's another example with the flower color. So this is our purple flower. It's big P, big P. This should be little p, little p. This is white. So what happens is the purple flower sends a purple gene, which is dominant, and the white flower sends the white version, the white allele, which is recessive. So all of their offspring have a purple, they look purple. But in their genetics, they've got a purple and a white copy. So what can happen is you've got a big P, little p for each of the parents for this next generation, and you make another Punnett square. And what you see here is 1 out of those 4 is a little p, little p. So that means their flowers would be white. So what you'd expect is there would be 3 purple flowers for every 1 white flower. And now you would have to breed a lot of plants to see this ratio. If you only did like 5 offspring, you wouldn't really be able to see a pattern. So in science, we need to make sure we do enough trials to make sure we get good results. And that's what Mendel found. He found 3 purple flowers to 1 white flower ratio. On our last slide, we've got a 50-50 chance. This would be a 1-to-1 ratio. We'd expect them to have an equal amounts of yellow and green pods. So let's discuss the difference between phenotype and genotype. Phenotype is what an organism looks like. It's their physical characteristics. So your hair color, your eye color, your skin color, your ability to roll your tongue, those are what we can see. The flower color is the phenotype. The genotype is the genetics. That's what alleles they have. So for example, for white flowers, the phenotype would be white. Their genotype would be little p. They have 2 white flower alleles. So you can tell an organism's phenotype by looking at them, but you don't always know their genotype. And the phenotype is based on their genes and then their environment as well. Mendel's principle of segregation. So one of his hypotheses was that those alleles, the different versions, separate when we make sex cells, so that the sex cells, the sperm and the egg, only have one copy of each. So here you can see this is the middle step of that monohybrid cross, the F1 generation, the parents, F1, and then their offspring. They have a 50-50 chance of sending out each of those versions. Only one gets sent out from each parent, and they can go to either. A dihybrid cross is more complicated than a monohybrid cross. So di 2. So here we're going to cross two traits. They have two characteristics that the parents are true breeding for. So the first generation parents, for example in this one, the seeds are wrinkled and they're yellow. And they have two alleles for wrinkled, two alleles for yellow. Whereas the others are round, they've got two alleles for that, and green. So what happens is they're going to send out a little r, big Y, big R, little y. So in that F1 generation, all of the offspring are going to look the same. They've got the big R round is dominant. It masks the wrinkled. And yellow is dominant, so they're yellow. And then when we cross this version, it gets a little more complicated. So our F1 cross, these offspring, these parents can send out different traits. You can have them send out both the dominants, the round and green, wrinkled and green, wrinkled and yellow. And there's a 1 in 4 chance for each of these. Just random. Each offspring is going to look different. Same with these. So what you have then is you have different possible traits. So out of these 16, you've got 1, 2, 3, 4, 5, 6, 7, 8, 9 of the squares have round yellow offspring. So that's what we'd expect in the 9 part for that. And then in our ratio, 1, 2, 3 would be round and green. 1, 2, 3 would be yellowed and wrinkled. And there's only a 1 in 16 chance that you would have a green wrinkled seed that would be sent all of the recessive. So if you gave 1,000 offspring and looked at the different traits, you would find this ratio, 9 to 3 to 3 to 1. Let's relate this back to homologous chromosomes now. So remember, homologous chromosomes are when we have two copies of the chromosome, one from mom, one from dad. So they have the same genes on there, but they can have different alleles, different versions. So those genes are at specific loci or loci. Those are the locations. And then you've got different allele versions at the same locus, at the same location. So here, that could be your trait for eye color. You've got one allele over on this chromosome, so that could be like for blue eyes, and that could be for green eyes over here. But it's the trait for eyes at the same location. So here we have another example showing different genes at different loci, and they're showing different possibilities. So this one here has two copies of the same allele. They're both dominant. This one has two copies that are both recessive, and this one has one dominant, one capital, and one lowercase, one recessive. So the names that we give for those, if you have two copies that are the same, you're homozygous, homo same. In this case, because you have dominant, you would be homozygous dominant. In this case, you are homozygous recessive. You've only got one version overall, but it's the recessive trait. And then when you have one copy that's recessive and one dominant, you're heterozygous. Hetero, different. So you've got two different versions, two different alleles. And in that case, you would only see the trait that's coded by a big B. So whether that be brown eyes, yellow seed pods that are round